So proteins have the 4 levels of structure. So we explored each of these during this chapter. Primary structure is the linear sequence of the amino acids within the protein chain. Secondary structure is going to be the regular and repeating spatial organization of neighboring segments of a single protein chain. So the 2 major secondary structural units that we looked at were the alpha helix and the beta pleated sheet. You should be familiar with both of these structural features. The tertiary structure then is the overall shape of the protein molecule. It's produced by the interactions of all the secondary structural units within the protein. So it combines all of these and it has the overall bending and folding pattern of the protein chain. There's also only a single protein chain that's involved in tertiary structure. So all peptides and all proteins have primary structure, secondary structure, and tertiary structure. Some proteins also have quaternary structure if they are composed of more than one polypeptide chain. So if you have 2 or more polypeptide chains coming into contact and forming some other unique structural unit, this is known as the quaternary structure of the protein. So some additional advanced protein concepts that we need to cover as well are these concepts of the native protein, a simple protein, and a conjugated protein. So we've got our overall protein structure, but there are a lot of components that get modified in our protein structure. A native protein is going to be defined as a protein with the shape in which it exists naturally and functions within living organisms. So this is how the protein normally would look like if it's working and functioning properly. A simple protein then is a protein that's composed only of our amino acid residues. Ribonuclease, that we've talked about now a few times, as our globular protein, as the protein that breaks down RNA, and we looked at the denaturation experiment with the ribonuclease enzyme. This is classified as a simple protein because it's composed only of amino acid residues. Some proteins then, in comparison to being simple, can be conjugated proteins. So a conjugated protein is a protein that also incorporates 1 or more non-amino acid units within its structure. So the conjugated protein may have a functional group or a small organic molecule that it might bind to that helps it do its functional activity. So both the myoglobin protein and the hemoglobin protein are going to be very good examples of conjugated proteins. They both contain the heme functional group. That's that small organic molecule that contains the iron residue that helps both of these proteins bind to oxygen. Both of these proteins would lose that ability if they were not bound to the heme functional group. So conjugated proteins contain extra stuff in addition to just the linear sequence. So our proteins then can be classified by their function. Proteins are really the worker bees within the cell. They have so many different functions. So they're probably the most interesting type of major macromolecule that you can study within the body. So we can classify the proteins based on their functions. So proteins can act as enzymes, and we'll talk about this in more detail. But ultimately, enzymes are going to be catalysts. They help mediate metabolic reactions. They help mediate the breakdown or the building of organic molecules within the body. So you would need enzymes to break down proteins during digestion. You would need other enzymes to build new proteins within the body. So the ribosome, where protein synthesis occurs, is actually composed of both RNA and protein that is acting like an enzyme to build that new protein structure. Proteins can also act like hormones. So what are hormones? We haven't talked about these yet in detail, but we will visit the very interesting topic of hormones. Hormones are actually molecules that are produced in one part of the body. They're secreted from that part of the body, and they travel a long distance to get to the site of action within the body where they're going to cause some effect. So insulin would be a very good example of a hormone that is produced by the pancreas. it's secreted into the bloodstream, and it travels around and it bumps into the liver. And in the liver, it tells the liver that it needs to pull glucose out of the bloodstream and store it for energy use later. So this is a good example of a peptide sequence that is also being utilized as a hormone. Insulin is a peptide. Some proteins act like storage proteins, so they'll make essential substances available when we need them. So for example, myoglobin would be a storage protein. It stores oxygen in the muscle tissue so that it can have extra oxygen available during times of severe exercise or stress within the system. So, you exercise and you start breathing heavier to try to bring enough oxygen to the muscle tissue. If you have more myoglobin, you're going to be able to store some of that oxygen for later use inside the muscle tissue. So proteins can also transport materials within the body. They can carry substances through the body fluids. We've already talked about serum albumin as well. Serum albumin is carrying fatty acids in the bloodstream. So this one is acting like a chaperone. It's going to block the carboxylic acid, very similar to how the coenzyme A does, except it doesn't do it in a covalent modification. It kind of just takes its own protein and wraps it around the fatty acid, and it hides that fatty acid from the rest of the cell so that the other cells in the body are not degraded or harmed by the detergent activity of the fatty acid. So serum albumin is a very good example of a transport protein. Some proteins also provide structural support, and we saw examples of these guys too. A lot of our fibrous proteins would fall into this type of class. So our collagen and our keratins are very good structural proteins that are providing a lot of support to the body. We also have protective proteins. These will defend the body against foreign matters. And so a lot of our immune system is going to be reactive due to these protective proteins. Immunoglobulins, or antibodies, are going to aid in the destruction of invading bacteria. So the antibodies that our body makes, or immunoglobulins, as they're called in their protein structures, are actually protein molecules that help recognize foreign things and get rid of them from our body. We can also have contractile proteins. These types of contractile proteins will actually do mechanical work. So two proteins that are involved in muscle contraction, for example, are myosin and actin. They're going to cause the contraction that we feel every time that we contract and move our muscles. This is controlled also by proteins. So proteins then can be classified by their function, and that's actually a good way to try to classify them, because you can put them into groups based on their activity. But we can also classify proteins in different ways as well. So in addition to function, proteins can also be classified by their interactions with other molecules. So there are a number of proteins that actually will get modified with different functional groups or different macromolecule components that become a core and critical part of their structure. So you can classify them based on what type of molecules that they actually interact with. So glycoproteins, for example, have a substituent or a carbohydrate portion that gets attached onto their protein structure. So these proteins are modified by the addition of sugar residues. So we can classify them according to their structural modifications. Lipoproteins are modified by lipids, so there's lipid portions that actually get attached to these proteins. So we'll talk about some good examples of both glycoproteins and lipoproteins as well. The high and low density lipoproteins are the major forms, the HDL and the LDL, that help transport cholesterol and other lipids through the body. So when you go in for testing to look at your good cholesterol and your bad cholesterol, they're actually talking about the LDL and the HDL. The HDL is the good cholesterol. So towards the end of the term, we'll get back into our lipid structures and look at lipid transport in the body, and we'll talk more about the HDL and the LDL systems. Metalloproteins are also very important. So we could call the hemoglobin protein a metalloprotein because it binds to the heme functional group, which contains an iron residue in there. And so that iron is a metallic component that will help that protein function. So any metalloprotein then is a protein that contains a metal ion. You can also have phosphoproteins. These are proteins that get modified by having phosphate groups get linked onto them. So remember we said that phosphate groups are very good at forming phosphoester bonds. They can do this with functional groups or alcohol groups on the amino acid residues. So amino acids like serine and threonine and tyrosine all contain alcohol functional groups in their R group module. So these can be modified or phosphorylated very easily. So our hemoglobin protein can fall into the metal ion proteins or the metalloproteins, but it also has its own class because it's a very important type or specific type of metal binding protein that also requires that small organic molecule called the heme. So the hemoglobin protein can also be classified as a hemoprotein. So our hemoglobin and our myoglobin would fall into these categories, and they're very important for both. I like how it says it transplants oxygen. It transports oxygen, so it's a transportation protein. And then the myoglobin is the one that's storing the oxygen within the muscle tissue. We can also have nucleoproteins. Nucleoproteins are going to be proteins that interact with nucleic acids like RNA and DNA. So these can be found like in the ribosomes. The ribosome, we'll see when we look more at protein synthesis, is actually a combination of both proteins and RNA structure that help mediate protein synthesis. So this would be a very good example of a molecular structure that contains nucleoproteins. So these are both different and very useful ways that we can classify our proteins. We can classify them based on their core structural features. We can also classify them based on their functions. And depending upon what we want to accomplish, both of these classification systems are useful and are widely used throughout the literature and research on proteins.